Looks like I should wait at least a minute or two for more people to join.
Yeah.
Okay, looks like most people are here. If a couple of people jump in a little bit, that's fine. We have a lot to get through. So I'm going to finish our discussion about proteins and proteomics and databases. Then we're going to end there with some questions, and I'm going to shift gears into a short introduction of structural biology and structural biology databases. So that's our ambitious agenda today. So last week we looked at this slide a number of times, so I'm just using it as a quick refresher and to get us all on the same page, because I'm going to, in the next many slides, get into experimental workflows of quantitative proteomics, and then we're going to talk about databases, and we're going to leave a lot of this technical detail behind, and we're just going to be talking about concepts. But it's worth, again, pointing out that almost all of the workflows or methods and database structures I'm going to refer to are, in general, using this same approach to collect data. And we talked about converting proteins into peptides and resolving those on a mass spec. And then, to refresh your memory, is this picture, really sort of a low resolution in terms of the detail that's really involved, of the first piece of data, which is an ionization and a detection of a peptide, and that's what these peaks are. And then those peptide ions are selected, broken down into pieces, and sequenced. And so these two major pieces of data acquisition are what comprise nearly all of our proteomic data. And where we left off last time was the discussion of how can you use methods or use this data to get quantitative information. And so this dense slide that I'm going to walk through briefly here is going to give you some insight into the next case studies that I'll present to you. And that's showing here, what I'm showing here, there are multiple techniques that enable quantitative proteomics. So one of the techniques often employed is what we call label-free. And the label-free will make more sense when we go down to these labeling concepts. But the idea is that you're taking many, many replicates. So in these pictures, I've got this tube that represents your sample, a cell, protein complex, whatever. And that's getting analyzed in the mass spec. One tube, let's say here, is 3 technical replicates. And then it's collecting all of this data. So replicate mass spec analysis for each sample is then all put together and compared in a quantitative fashion. And this is what I'm calling indirect quant, because you're really deriving your information from each sample separately and then comparing them to each other, but indirectly. So these are not being acquired in the same mass spec run. But I'm introducing here labeling techniques which solve that problem, and that allows you to do direct quant. So you're actually measuring 2 samples at the same time. And so there's a metabolic labeling technique, which I'll explain in the next slide. But for now, let's just call it a label. That allows you to label 2 samples differently, combine them, and run them in the same mass spec run. So now you get to quantify one versus the other, and that's a direct quantitative comparison. And so this is an example of multiplexing two samples together or more, and then you can run fewer experiments, but you get more quantitative power out of this, okay? So I just gave you the super simple idea of how can I convert 4 different samples into two measurements. Then we have a barcoding technique, which I'll explain in a little more detail in a few slides. But nevertheless, imagine that you're barcoding each sample, mixing them all together, and then you're taking one measurement and comparing all the samples to each other. So this is increased multiplexing and direct quantitation. So I'm going to walk you through case studies of each of these techniques. But first, I wanted to give you a little more detail about the metabolic labeling, which the technique's now been around for quite a while, but it was really the first time that we were able to directly compare mass spec samples and get true quantitation from very ambitious large-scale proteomics experiments. And the technique is actually beautifully simple. All that's done is that you introduce a heavy atom, so in this case, carbon. So all you need to know is there's a light and a heavy. Okay, so the light sample has normal lysine, and that's carbon-12. That's the most abundant form of carbon that naturally exists. But then you can also get a isotopically enriched heavy lysine. So that means that your carbon is carbon-13. And the trick there is the carbon has, if you compare carbon-12 to carbon-13, it's 1 Dalton. Okay, so if you have a lysine molecule that has 6 carbons, your label introduces 6 Daltons, or let's just call them 6 mass units. And why does that matter? Because if you think about our introduction to this technique, a mass spectrometer just measures mass, correct? So you have a peptide of a defined mass. If you take that same peptide and you label it uniformly, and each lysine is 6 Daltons heavier, that means the entire peptide is heavier. So this is one example of introducing a label that is specifically resolved by the mass spectrometer, but can be applied to every single protein, every single peptide in the sample. And so when we talk about other labeling techniques, it's still that same concept. You're introducing a heavier molecule so that when you measure a peptide and its mass to charge, the exact same peptide from a different sample is now heavier. And so I illustrate that here. If you think about these red cells are producing peptides in red, the blue cells are heavy. They represent peptides in blue. If you mix them together, they're separated by, let's say, 6 Daltons for a simple example. Now you have 2 peptides separated in mass, and you can sequence each of those, and then you can actually derive quantitative information. And I'm showing you schematically here. Imagine if you had increased abundance in the second sample, you could directly measure a ratio or change in that abundance between the 2 samples. So a very simple technique turned out to be extremely powerful for introducing quantitation into proteomics. And so this is a busy slide. Again, it's a resource slide for you. There's a really nice review that I'm referencing here that gives you a detailed methodological breakdown of many types of ways that labeling can be used. I'm pulling out this first panel from figure A to show you this in a little more detail, this barcoding example, where in this case, 10 different heavy labels are introduced to peptides. So don't worry about all the details in the chemistry. Think about the example I just showed you before, where you had a uniformly labeled proteome of one type and then one mass unit heavier. Now imagine that you could have 10 discrete mass units that could be applied across all your samples. So when you mix all these together, you could have 10 samples that all sort themselves out by 10 divisible mass units. So it's a very simple barcoding strategy. And I use the term barcoding because it should remind you, I hope, of some of the things you learned about when you sequence genomes or transcriptomes. So barcoding is used all the time in next-gen sequencing for nucleic acids because you can add little sequence tags to your sample, mix them all together, next-gen sequence the whole lot, and then you could pull out that information after you do the sequencing. This is the next-gen multiplex barcoding for proteomics in a nutshell. Okay, so there's more details in the paper if you're interested. So I'm going to return here in this example from an earlier paper from 2006, but this is still considered one of the major breakthroughs and sort of a classic piece of literature in the field about the first application of a labeling strategy to understand proteomics in a quantitative way. And this study in particular looked at how cellular signaling pathways could change over time in the presence of different ligands. So here, we're not going to go into the biology. I'm just going to show you this is a broad strokes example, but they were examining the EGFR receptor pathway, where you could take a very simple experiment where you had cells, you could add EGFR, and then you could ask the question, how do all the phosphorylation sites across the cell change after 5, 10, or 1, it was 1, 5, 10, 20 minutes of EGF stimulation, okay? And the way this was achieved, and it's depicted here, is you have 3 different conditions, and each cell was each cell line was heavy labeled in a different way, and they used clever combinations of this metabolic labeling, which I'm just distilling it right here in this little picture from the paper. Is here are mass spec triplets or peptide ion triplets. So these represent the exact same phosphorylated peptide, but in 3 different conditions. So in this condition, there was no EGF stimulation, then 5 minutes, 10 minutes, and I hope you could appreciate that there's a quantitative increase in phosphorylation. Okay, and so now imagine this on a whole proteome scale where they measured tens of thousands of these phospholipeptides and assembled the entire architecture of signaling. So flashing forward to 2019, and this study that I chose happens to be the same investigator. So this is Jesper Olsen. Now in 2019, he has his own lab, of course. He led the group that did that first big metabolic labeling experiment, and they've published quite a bit in this field, but this is taking an example of, where has the field progressed in 13 years, still using a lot of the same fundamental ideas. You're introducing labels, you're using modern mass spectrometers that are faster, they're more accurate, and it allows you multiplexing in ever greater and greater and greater complexity. And again, this is a very busy slide, and it's here for your reference if you want to look at this paper. But you can see the scale that they're now carrying out these types of experiments, where they're looking across 4 different tissues and using high throughput methodology to do arrays of samples. And again, introducing labels so they can measure each one, quantify, and bring back quantitative information. In this study, they were looking at how these phosphotyrosine signaling pathways change depending on the environment of oncogenic mutations. So now, as I said the last lecture, we're taking the increased sophistication in the mass spectrometers and the workflows, so this combination of technologies, to start gleaning greater biological insight using quantitative proteomics. And this is just one of many, many examples. Another one I thought that was very interesting is thinking about how phosphoproteome, that when I say phosphoproteome, that means all the sites of phosphorylation in a proteome. So it's beyond just asking the question, what proteins are expressed in a cell or an organism, or the question, what sites are phosphorylated, and now asking the question, how do all these sites change in a biological context? And this experiment was to look at how the phosphoproteome of the rat or mouse changed over time in an effort to try to understand how much signaling is dependent on a circadian rhythm. And it's an incredible study, again, employing many of the techniques that we walked through, but they showed that close to 25% of all the phosphorylation events in the rodent liver are responding to different times of day, giving us tremendous insight to how maybe circadian rhythm is controlled in a whole organism in a tissue. Okay, so that was the very high level, very short and broad overview of quantitative proteomics. As I gave you in my disclaimer the other day, I'll say it again. Each of these papers and each of these topics could really be their own lecture, but I was just trying to give you representative studies to take you from the earliest form of quantitation to where we are in, today in 2021. But I think even more relevant for this course is spending some time focusing on database structure and how all these studies in the literature and other places are being pulled together and are available for data mining exercises. So an example that I like to point out is comparing the state of the human genome to the state of the human proteome. And I'll thread this example from the literature into database examples, but for me, for this lecture, I like to point out that there are big differences from where we were in 2001 with the human genome sequence to just over 5 years ago with what was considered the first draft of the human proteome. Nevertheless, these are major landmark studies. I don't need to go into all the aspects of the human genome sequencing, but I'm sure based on your other lectures already in this course, you can appreciate a lot has changed in 20 years. Proteomics lags behind. And I'm going to discuss in the next couple slides and point out, again, this idea that we're nowhere close to the same coverage of the human proteome that we are of the human genome. And the idea is pretty simple. For argument's sake, let's say that we have many, many times over sequenced every single nucleic acid in the human genome. And now we've done thousands or hundreds of thousands of human genomes. That is not the case for the human proteome. Okay, so these articles that have the draft map, this is not a complete map of every amino acid that can account for every open reading frame in the genome, okay? Nevertheless, these are landmark studies because they are a draft map of potentially every possible observable or visible protein that exists in a human, all right? So it's equivalent to the human genome, but in protein space. So what do some of these data sets look like? Again, a very dense slide, but what I want to focus on is, and this comes straight from the paper, and I've put the references down here, but what you're really getting is a collection of, again, many separate mass spectrometry experiments. So they're taking, in this study, they were culling from public data repositories, so labs that either collect data on the human proteome, deposit that, or different publications. So they pulled in, there's a lot of numbers here, but I'm just going to call them experiments or runs, so a single run of the mass spec. So they took 10,000 experiments from the literature or databases, then they performed their own 6,000 experiments with a simple goal: try to account for every protein that should exist based on the genome and map direct evidence for that in the proteome. And so the coverage is shown here, and the percentages imply that the coverage is pretty good, like here's 93, 95. And that means that they can account for, on each chromosome, let's say close to 95% of the protein coding content of that chromosome. But again, keep in mind, this is not full amino acid resolution. Some of those proteins, they may only have a handful of peptides, right? There may be proteins that themselves are only 5% covered, but nevertheless, you can say, okay, I have really good evidence that that protein is expressed, therefore I'll call it covered, okay? So keep that in mind. But one exercise I like to carry out is, why did it take us so long? If the genome was sequenced in 2001, why did it take so long to assemble, over 13 years to assemble a proteome draft? And it goes back to the statement I made about how mass spec technology has really driven the field. So the question is, what if you did this project in 2001? Okay, took nearly 6,000 measurements on your own in your own lab, okay? In this case, in the lab of Bernard Kuster. If they used 2001 technology, it would have taken them 61 years. So you can already do the math. If they had started and kept on that course, they wouldn't be done, okay? But in 2014, which we can estimate is about when they started working on this story, using 2014 technology, it took them a little bit less than a year, okay? So again, as I said before, the pace of the field is really dictated by the technology. As mass specs got faster, more accurate, you could think about doing 6,000 experiments in your lab, and you wouldn't have to convince someone like a poor graduate student or postdoc to spend 61 years doing it, right? Okay, so that's a powerful example of why did it take so long? Because the technology had to mature, right? So there were two papers that reported this draft map, and the second paper did a lot of the same thing. They took existing data and they also collected their own data. But I like this slide because it points out something important. Unlike the human genomes, which are typically derived from, let's say for argument's sake, from a single cell type or a representative genome sample that's in all the human genome, the proteome is not the case. We have collections of data sets from all different types of tissues, from lung, heart. It's all illustrated here. In this study, they took many clinical samples. They took cell lines from their lab. They did some experiments where they differentiated hemopoietic cells. But the take home is that the proteome as we know it, so if we map this, if we have a database that says this is the human proteome, it's a collection from many other tissues, because every cell and every tissue has a different repertoire of protein expression. So if you just focused on, let's say, one cell type, you wouldn't necessarily get the same comprehensive map of the human proteome, because protein expression is dictated differently. So the concept here is we'll collect many samples representing many different proteomes. They all map back to a human, but as you can imagine, this comes from many different humans and many different samples. So it's not a single human's proteome. It's a entire human proteome, as sort of a collection of representative proteomes. So databases. One of the still very successful types of database is actually a peptide database alone. You can always go back and understand where were these peptides derived from, but I think it should be very clear to you now that the proteomic databases are intimately tied to the peptide level information that's acquired in the mass spectrometer. So it's important to have databases of that peptide information. And so this is just one example. Again, a lot of words on the slide, a lot of links, but it's just to let you know that there are databases that really focus on just the peptides. And that can be useful because let's imagine that in a database, so let's take the human database, for example. I just told you that we don't have peptide level information for all the proteins in the humans, but there's sometimes a lot of redundancy. So you can imagine that we have one or two peptides that may have been represented in every single experiment. So you could start to ask questions of how do data sets, or how do peptides compare across data sets, either the ones that were observed very frequently or those that were observed less frequently. So these are very valuable resources for the community. And there are a lot of different ideas out there, and there is some effort to sort of bring all the ideas together in how should we have a standard for indexing and reporting peptide data and proteome data. And so there's some links to that literature, so you can see how the field is still thinking about it. But the take home for you is that it hasn't been resolved. There really isn't one way to deal with proteome data. There are many separately funded initiatives, and there's some connection between them, but you should really keep that in mind when you're doing data mining and you're thinking, where's the source coming from and how connected are those sources? And what is the completeness of that database archive? So there are also specialized peptide and protein databases. So I'm drawing a connection here back to one of the Human Proteome articles. And this site, still being curated today, is really they're sort of this group's in-house curation of human protein expression and mapping through proteomics. So you can get lab-specific, organism-specific databases. And again, the word of caution here is they're not always coordinated. This isn't necessarily going to be directly linked to, let's say, a national resource in the United States. They maybe connect to other countries and other types of virtual data warehouses. I did mention that there are efforts to try to tie everything together. And this busy slide is giving you one example. That's a little bit outdated, but it's pretty much still the same infrastructure. This proteome exchange, and the idea is we recognize as a field that there's lots of ways that data are collected and then curated, so we can have one central resource that tends to pull everything in and keep it together. So you can always have a uniform way to curate the data. It's similar to depositing genome information that's being curated, let's say, in United States government databases. So it's called a proteome exchange. And the idea is you can upload your raw data and they will curate it and it'll be there available to the community. And it will also help tie and overlap with other types of popular databases. So this has actually been quite successful, and it's still maintaining popularity and activity. So we already talked about this slide last time, but I wanted to put that in there, that in addition to, peptide level data, protein level data, there are protein-protein interaction databases that we talked about. And so this, if you recall, this can be connected to the raw data from the mass spectrometry, but often it's just simply reporting these two proteins were in complex with each other, or this one protein had a complex with 50 other proteins. Sometimes you can get quite quickly, in fact, separated from the raw data that actually contributed to an individual interaction, but nevertheless, the interactions are curated. This is the short section of the lecture where I point out that there are other databases that give us very important information about human proteins that do not use mass spectrometry. In this example, this is what's called the Human Protein Atlas, and it's actually based on a very labor-intensive, carefully curated set of antibodies. And so, as you should know, that antibodies can recognize proteins quite specifically, and you can label a sample and produce a signal, and that signal tells you your protein's there, and you can learn where your protein is. So I'm comparing here two studies, one that I mentioned very briefly done in yeast. So this is a classic study where a green fluorescent protein was fused to every open reading frame in a yeast cell. That means if the protein was being expressed, the yeast would glow green. You could take a picture of that and you could see, you'd answer two things. Is the protein expressed? Yes or no? Where is it expressed? And so you can see all the different localizations. Here they're showing you like the nucleus, the mitochondria. So here we have proteome level information without a mass spectrometer, and you're starting to learn more of the biology and perhaps biology that might be difficult to get from a mass spec. So the same type of workflow was used to characterize the localization of around 13,000 human proteins, but instead of gene fusions, they used antibodies. So here we're having basically doing lots and lots of immunolocalization studies coupled with imaging to curate the expression and localization of 13,000 human proteins. And then, as this paper showed, you get a subcellular map of the human proteome. So now you're dealing with expression and localization all without a mass spectrometer. Nevertheless, a very, very powerful proteome level database. So here I just want to briefly point out the state of the art for single cell proteomics. And this is important because, as I'm sure you're aware, genome and transcriptome level studies have broken the single cell barrier. And of course, there's ongoing efforts to start to analyze single cells at the proteome level. But the bad news right now, if you don't know it already, is that we have not even We haven't achieved that. We're not even close. So I'd like to bring this paper up. It's a fantastic paper from Jonathan Weissman's lab. It was back in 2006. And if I tell you, oh, we're not at single cell resolution, you might look at this title and say, but didn't Jonathan Weissman give you single cell proteomic analysis of the entire yeast? The answer is yes, but what they measured was a single protein in a single cell, but they did it one at a time. So they did 2,000 plus cells one at a time, but it wasn't a single cell that gave us information about 2,000 proteins. So you can see the distinct difference. There, in this study, they were looking at single cell information about protein expression, but to get at the 2,000 to 3,000 protein level, you had to image, again, 2,000 to 3,000 cells. So that's not exactly the same as single cell proteomics, where you should be imagining one cell and all of the protein content of that one cell, okay? So there are examples in the literature. Again, here's a busy slide, and I'm not really going to go in too much detail, but there are technologies. The one at the top, CyTOF, it does include a mass spectrometer. And then there are other technologies like this single cell capture technologies that are beginning to approach getting protein level information from single cells. But even though these techniques are very, very powerful, they're for a small fraction of the proteins in a single cell. So CyTOF, I think it's on the order of 50 to 80. It's probably, maybe it's even larger now. They've introduced more and more labels. Very powerful technique, but you're still only in, let's say for today, maybe a couple hundred proteins. That's far short if you're trying to think of a cell like a human cell that may have 30,000 proteins. Or as we discussed, if you include protein modification, 300,000 or some estimates up to 1 million different types of proteins in one cell. So the resolution is not even close. But nevertheless, there are powerful answers to single cell proteomics that use capture technologies and labeling technologies. And again, I like to bring up this point. We talked about it earlier, but we have a sample limitation problem. So we don't have a polymerase. So single cell genome and transcriptome analyses are still giving you access to amplification techniques. You can take a single cell and you can amplify the DNA. You can't do that with proteins. You cannot amplify that signal. You can't take the proteome and generate millions or trillions of copies of it to increase your detection. Because proteomics has no polymerase, we don't have an amplification method that can do that. And the mass spectrometry has limitations. There's no single mass spectrometer that can provide a complete cellular proteome, and certainly we're not to the point where we can take, let's say, a single cell and shoot it into the mass spec and see all the proteins that are there. So I just put this again as a reference about some of the challenges. If you're interested, you could read about some of the thoughts of where we could break that single cell barrier in things like sample preparation and different ways to manipulate or advance the technologies. And again, this is a busy slide, but there is a technique that's gaining popularity, and it works quite well, of again using these barcoding strategies to quantify single human cells with standard quantitative proteomics workflows. But the reality is shown here on this graph. And the only thing I want you to notice is that on this axis are the number of proteins. Okay, so this top limit is, let's say, 1,000. And so in these bins, let's say in one bin, you have only 1,000 proteins. So to get to these levels, right? So let's imagine that this is a good fraction of a proteome. You still need 10 to the 7th cells. So this is even to get, and this isn't a complete human proteome coverage, you still need many, many cells. So when you do the same technique and you look at single cells, you can see the greatest bin here is 200 proteins. So it has a lot of promise. There's certainly an achievement in this technique, but it's not single cell proteomics even close to what you might think if you did a single cell transcriptome. So it's something to think about very carefully and keep in mind that the field is advancing, but it still has a long way to go. Okay, so I did a pretty good job staying on time because I'm going to spend the rest of our time talking about structure, but I want to entertain questions now about anything in the first two lectures. So it would be a good time if you have a question now. It's okay, you can ask me again at the end, but I just thought I'd take a quick pause here. Questions about quantitation. What is a mass spec? I hope not. No. Silence. Okay. All right, then I'm going to switch lectures here to structure. Okay, so at this point in the course, you've had very fast and information-rich discussion of the origins of DNA, RNA, and protein data. Later in the course, you're going to do some modeling, and you're going to think about protein dynamics and how proteins move. And so it's important to get, a very high-level sense of how is it that we have databases full of protein structural information, and where does that structural information come from? So I'm going to give you a very basic and quick introduction to X-ray crystallography and cryo-EM. First, and you can already have access to these slides, so for later you can go back and look at them. First, I'd like to point out that on these topics, there are complete courses available to you for in-depth discussion on cryo-EM, structural biology, and far more details and technical information about these two fields. They're very complicated fields. There's a lot of information, and there's no way I'm going to give you that depth and breadth in less than 45 minutes. Okay, so I listed some of the available courses here, a couple tutorials and reviews that are really useful if you want to think through any of the methods we're about to discuss, and you want to educate yourself a little more on some of the technical and perhaps the mathematical details, because there's no way I'm going to be able to get into all the mathematics and biophysics and physics that are behind these techniques. So the first thing I like to start with is the why do we need these techniques, right? Why are there fields and technologies and complete disciplines and departments in universities and all over dedicated to this information? And one very simple thing is that it's impossible for us to just see a macromolecule. Okay, so as you know, our eyes use visible light, and even microscopic techniques, especially light microscopy, uses light. But the problem is that there's a limit to how small you can see things with regular visible light. Okay, and so the concept here is the diffraction limit. And the concept of diffraction will become much more clear in the next slides, but the point is very simple. If you're trying to see something that is, so here's this perfect, nice little picture of the backbone of an amino acid. If you want to see a carbon-carbon bond, right? If you want to understand the position of the molecules, the atoms in a polypeptide, in a nucleic acid, you need to have a diffraction limit that's below 1.5 angstroms. So that's the 1.5 is the distance between a carbon-carbon bond, okay? And that's just not possible with visible light. So you need to have X-rays, okay, to resolve atomic features, because listed here are the wavelengths of different types of X-ray sources. We're going to talk about that in a minute. So this might be, I'm jumping ahead a little bit in some of the nomenclature, but just think about what is emitting this X-ray and what is the wavelength. We can have a lab source, or we can have a more powerful source of X-rays, and we can get wavelengths that are below where you can get them below 1.5 angstroms. So that means they're smaller than the distance between carbon and carbon. So that gives us the possibility to actually see at atomic resolution the atoms in a molecule. Okay, and so a simpler way to think about this is, and sort of, I love this figure because it maps all the different types of imaging technologies according to their resolution. And so up here you have from a mouse down to a small molecule and everything in between. And all of these techniques are using, most of them, visible light to determine things that are as big as a mouse all the way down to the size of a virus. And what's important here is even with what's called super resolution microscopy, so techniques and technologies that get us very, very small. Of course, you can appreciate that probably from a hair on down, you need a tool to see, it's very difficult to see a single cell with your naked eye. So you need imaging technologies, and these are all very powerful imaging technologies. They're not even close. So this is a nanometer scale, and 1 angstrom is 0.1 nanometers. So you need to be at 1.5 angstroms. You're not even close, even with super resolution and basic electron microscopy. Now we're going to talk about cryo-EM later, but even with standard electron microscopy, you're not getting low enough to see at molecular detail. So there has to be some other way to visualize molecular structures. And so that was with the X-ray. So this is an incredibly overly simplified view of how an X-ray works, or allows us to see at atomic resolution the structures of proteins and nucleic acids. So this image pictures an X-ray hitting an atom and producing a diffraction pattern. And so we were just discussing this concept of what's the resolution limit, and we have now X-ray sources that can diffract at resolution that's below the distance between two atoms. Okay, so you're in the very simple setup, you're bombarding your sample with X-rays, and then you're producing a diffraction pattern. So you're getting direct detections of atoms, and that's what's important. This is a direct detection using an X-ray to directly detect the atomic composition of the material you're interested in. And crystals are required, and we're going to talk about that. So that's right there, it tells you the name of the technique. It's X-ray crystallography to determine the structure of proteins and nucleic acids. Now, there are other methods that we're not really going to go into except for cryo-EM, but this is the basic one. And at the end of the lecture, it'll become crystal clear, why am I spending so much time on X-ray crystallography in a database structure course? So just thought I would show you, here's a diffraction pattern, right? So that's as simple as it gets. If you, in this experiment, someone took a crystallized protein sample and shot it with X-rays and revealed a diffraction pattern and solved the structure. And so you should immediately realize that there's got to be a lot more to it than this, because this isn't like reading the output of a next-gen sequencer or even a mass spectrometer. This is a very cryptic and bizarre puzzle that you need to solve before you can translate this. This is the raw data from a diffraction experiment. How the heck do you get a crystal structure out of that? So unpacking it a little bit, why crystals? Crystals are special because they are, by definition, a very exact order of atoms. So to get a crystal, you need to have a lattice, you need a repeating element that grows and grows and grows. So crystals are macroscopic assemblies of microscopic pieces that are all exactly the same. Okay, so the crystal by definition is required because you want an order of the molecules in exactly the same way, in exactly the same position in space. Okay, and I'll show you exactly how that translates to a structure in a minute, but this is also a really important slide because it allows you to think about where does the information come from in a structure, right, in a protein structure. And here's the basic workflow for determining a protein structure. This workflow is minus some of the details of crystallography, but also similar for EM, for cryo-EM. You have to generate the protein, express it, purify it, and then you do all this crystallization. And what I like to point out here is we're not, for, I would say, 99.9% of the structures in the database, we're not taking native proteins out of a cell. We're not taking a cell, opening it up, crystallizing it, and determining structures. You're taking a single protein, a single clone, the DNA level, and amplifying that protein, usually in a host, so human protein being expressed in bacteria, in cells, in bacterial cells, or in yeast cells, or in even insect cells, to get lots and lots and lots of that protein, and you purify that to homogeneity. So you're increasing the copy number of that protein, and you're purifying it, so you have just that protein. So the take home here is it's rarely a native protein from its native source. You bring it into a system where it allows you to amplify the amount of that protein you have for your experiment. So that's different. Going back to, the core of this course, that's different than you could sequence a human genome, and I could sequence my genome or your genome from us. And right now, I can't think of a protein that I could actually isolate from myself and crystallize. I would have to take a representative gene and introduce that in some sort of an expression system and carry out what eventually is a very long, detailed process of biochemical process to result in a protein structure. You still get an understanding of what that protein looks like in three-dimensional space. But what I want you to remember is that it's not a native protein. There's a lot of tools and techniques that get us to that structure. So we're not determining the structure of real human proteins from real humans. Okay, it's derived, all right? Data collection, this is just, if you've never seen something like this, I wanted you to be able to see it. Literally, this is the apparatus. Now, this is a very simple view of it, but you have an apparatus where you hold or mount the crystal there, and then you put it in front of a X-ray beam that shoots the crystal and ablates it, and then you get these diffraction patterns. So this is exactly what it can look like in a laboratory source, and there's even similar apparatus that work at these big synchrotron sources, which I'll show you in a minute. So X-ray sources are important. So as the first slide, I showed you that the difference between 1.5 angstroms and 0.5 is a lot, because 1.5, let's say you can generate that in your lab. So there are sources at Yale that you, in the med school and on Science Hill, that you can generate this type of X-ray, but it's not going to be high enough quality. It's not high enough resolution to really see atomic detail that you want. It's a good starting point, but to get the really, really high quality data, you want to go to a synchrotron. And so these three pictures here are, these aerial view, these drone shots of these synchrotrons. And these are 4 of the more popular ones in the United States. What these are particle accelerators. So these are these massive underground tunnels where physicists accelerate atoms of all types around and around and trying to understand the fundamental composition of matter. What's really nice about these setups is that they produce X-rays, and they produce really powerful, consistent X-ray beams. And so this picture might be a little hard for you to see on your laptop or whatever, but you can kind of see the circle with these little lines shooting off of it. And what that depicts are different types of beams, so particle beams and sources of energy that are being extracted from the accelerator. And a lot of these are X-ray sources. And so a side product of these cool experiments is really nice, pure, powerful X-rays. And so what happens is structural biologists from all over the world mail their crystals to these sources, and then one at a time, either with a human, or now many of these are automated, and there's robots that'll load up your crystal, and you can use these really powerful, very high resolution X-ray sources to blast apart your sample and get really high quality data. So this is where the work is really done. The majority of all the high resolution, high quality data comes from a handful of these synchrotron sources all over the world. And I like to point out that X-ray diffraction has a long history and a lot of fame in the field of biology. So this is actually an X-ray diffraction pattern that's very famous because it was the X-ray diffraction pattern that Rosalind Franklin produced from DNA. And so it wasn't a crystal, it was actually a thread of DNA that she could extract and again bombard with X-rays and generate a pattern. And it was from this information that she and others deduced the primary structure of DNA. So a little bit about how is this pattern produced and what's the connection between a diffraction pattern and a crystal. And so, as I mentioned, the crystals are important because you get, by definition, a order of molecules that have a position to one another that's fixed. And within that fixed position is a replication or an expansion of, in crystallography, we call it a unit cell. So you have this unit cell that's repeated over and over and over again. And what that does is it gives every single molecule in your sample the same position. And then what that does is it actually amplifies the diffraction pattern. It wouldn't be enough to just take one molecule and shoot an X-ray at it. The power is you have thousands, maybe millions of molecules all lined up. And so the diffraction pattern amplifies itself and allows for these spots to form. And this is the point where I'm going to blow past a ton of detailed information in mathematics. But because we know that crystals form a predictable regular pattern, and we have a good sense of what those patterns are. And so here's an example of how a water molecule might line up in a unit cell. So there's two water molecules facing each other, and this is the way they're going to look throughout the entire crystal. Okay. Imagine that's true for a protein, right? A protein is every single protein molecule is lined up in the same way relative to each other. All you have to do is figure out in what way they're organized in the crystal, the lattice, and you want to understand the dimensions, these unit cell dimensions. You can derive that information from the diffraction pattern. And then with a couple other measurements, you can turn this diffraction pattern into the exact location, the exact three-dimensional location of your molecule in the crystal sample, okay? And so, and that's basically it. If you can tease your protein into crystal form, and you can collect a high-quality diffraction pattern on a high-quality X-ray source, you could derive the three-dimensional position of your molecule in that sample. And one of the things, what you actually produce is what's called an electron density map. And so when you're diffracting X-ray sources off your sample, you're learning where in the sample are the electrons located. So in atom by atom, where are the electron dense regions? And so those map to the pieces of, that's in this example, the positions of the R groups and the backbones of amino acids. So this is really key. You first make an electron density map, and that tells you this is where the molecule is positioned and how it's positioned in three-dimensional space. And this is real data. This is the actual raw data from the experiment, transformed from electron density, or transformed from the diffraction pattern into a real electron density map. Then you have to build a model, okay? So you have to figure out how do the amino acids fit into this model. But here's the great, cheat or solution to this whole problem. Just like what I said with mass spectrometry, we're not in proteomics. We know we know the sample that we have. We know what organism it comes from. Sometimes we even know the proteins that should be there. When you've cloned, overexpressed, and purified a protein, you know what the protein is, and you know the composition of amino acids. You're not trying to map every possible combination of all amino acids in every organism on the planet to an electron density map. No. You know, you have a protein that you've isolated and expressed, you know the sequence, you know where it starts and where it stops. You actually have it as a linear chain. And so what you're doing is using computational methods to thread and match your amino acid sequence to the electron density map. So you're not trying to derive all this from nothing. You have a lot of information. Once you produce the map, you know what the molecule is that's in the experiment, and you just have to fit that. Okay, so you measure the map and then you build a structural model. And this example highlights why resolution is so important. Because what I just said is that you know what your sample is, you know what you've crystallized, and you have the amino acid sequence. But what this shows you quite easily is the difference between an electron density map that's only at 3 angstroms resolution versus 1.2 angstroms. This blob is a phenylalanine. And so you know right away, like, how confident are you that if you map your phenylalanine here, if your model says that's phenylalanine, okay, maybe. I mean, because this is like a 6-carbon ring, it's probably going to only fit phenylalanine. But you certainly feel a lot more confident when you have 1.2 angstrom resolution, you have a nice hole in the middle because there's no electron density there, and you can see your 6-membered ring right there, clear as day. You see each carbon atom with a nice electron density cloud around each one. Very high resolution data allows you to fit your model with extreme accuracy. And here's one of like the highest resolution structures ever captured. This is at 0.4 angstrom resolution. So you can see here the absolute certainty you are that you've predicted your model is fitting the electron density perfectly. Okay, so that's I've opened up the fire hose of information here, but take home this, that there are thousands and thousands of structures, but they are not all at the same resolution. And you have to think about how confident are you in the picture or the assignment of that 3-dimensional structure if they're all not as high quality in terms of resolution and model fit in the database. So that's something to keep in mind. Resolution really matters in getting what we think is the accurate model in 3 dimensions of a protein. So what happens when you achieve this type of workflow, you get this type of resolution, you can start to get access to the biological information that you're interested in. So here's a famous example. It's still a beautiful structure. This is a nucleosome at 2.8 angstroms. It's not sub 1.5, but nevertheless, enough resolution that this model could tell the difference between nucleic acid double helix and the protein core. And you could see that pieces of the nucleosome shoot out and extend beyond the DNA bound around it. So if you don't know what a nucleosome is, right, that's the fundamental unit of chromatin, right? You have proteins that organize the genome by wrapping DNA around them. And this was the first structure of that complex where you could see base pair resolution. So you could tell the difference between A, T, G, and C. See, these are the electron density maps to those base pairs. And you could differentiate a nucleic acid structure from a protein structure and separate out histone from DNA, okay? Biological information. Another famous complex of proteins and nucleic acids is the ribosome, which was solved here at Yale by Tom Steit's group. And this work was so important that it won the Nobel Prize in 2009. And this work allowed for 2.4 resolution of the entire ribosome, the large subunit, and you could tell the difference between the RNA components and the protein components of this massive molecular machine. So structural databases in the last 20 minutes of our discussion. So by far the most popular and curated and supported one is the protein database. This number is a little bit out of date, but just like any database in active fields, it grows and grows and grows. So there's approximately 1,000 or 150,000 structures and growing. It has an awesome data. There's a lot of information on this database. So you can, spend many hours walking through its different links and explanations of different types of macromolecules. But what I like to point out is thinking about where do they come from? So if all those 100,000 structures, it's heavily biased towards human. So there are a lot of funding agencies and a lot of desire to try to understand the structural component of every protein in a human. So by and large, the most structural information we have is from human proteins. and that's going to continue on and on. There's more human protein structures deposited than any other organism. So keep that in mind. This is a organism bias database. You're not going to get the information for every possible protein in nature. We're going to be biased towards human structures. And I'm going to show, when I switch to EM, I'm going to compare these, but by and large, in the structural database, at least the PDB, it's mostly X-ray crystallography. That's still contributing the most structural information. And those resolutions range from 15 angstroms down to 0.4, right? And I would say the majority are somewhere in between that, right? So they're not all sub 1.5 angstrom structures. Most of them are greater than that. So that's something to keep in mind, that even though there's hundreds of thousands of structures, the resolution is not the same, okay? And EM, it gets a little worse. We'll discuss that in a moment, but that resolution can range from 70 to 1.8. And so the majority of the EM structures are greater than that 1.5 barrier. There's very few EM structures of proteins that are really subatomic. So that's important to keep in mind. And you can also think about, where do the proteins come from? So most of the proteins that we understand are soluble proteins. Far fewer crystal structures are of membrane proteins, for example. These are notoriously difficult to crystallize. So there's limitations to the methods and the technology that gives us access to those structures. So that's something to keep in mind about what is in the structural database. You're not getting as much information about membranes as you are about the soluble parts of proteins. And there's not as many complexes, right? So mixed protein nucleic acids is not nearly as much as single proteins on their own in solution. So it's important to keep that in mind as well. Like what do we really know? What's the depth and the breadth of the structural information? So briefly, we're going to close in the last 10 minutes on cryo-EM. Phenomenal advances before 2015, but certainly since 2015, has really resulted in an explosion of the technique. It's starting to almost keep pace with X-ray crystallography. So it's important to note. And it was awarded the Nobel Prize in Chemistry in 2017. So it was recognized that it was really a major breakthrough, and it's accelerating our structural view of macromolecular complexes and biology. So as we discussed in detail, we have a crystal that we bombard with X-rays, get a pattern, and then we figure out a density map. And in crystals, we need a lot. So, somewhere around 10 to the 12th copies of a protein are required to really get a good structure. The nice thing about cryo-EM is that the barrier is lowered, so we don't need as many. So somewhere 10 to the 5th, and this probably has changed. But this is data from my colleague, Fred Segworth. But it's interesting because you need less material and you don't need to crystallize. Now, there's many, many details about the source of the sample, and we don't have time to get into it, but just a simple take home is that the possibilities are much greater. You don't have to have crystallization, so you can sample from membranes, from fewer molecules, from more native-like conditions. So that opens up far more possibilities. The term would be access. You have access to far greater biological samples in potentially in EM. And a lot of that has been borne out in the literature. So it's exciting because it's an alternative to crystallography. It gets around some of the major pitfalls and limitations of crystallography, and it's really proven to give us a lot more insight into biological structures. So in a nutshell, in a cryo-EM experiment, and cryo is because you're freezing it. Again, this is some of the details that we're going to have to gloss over, but you're doing a very low temperature preservation of your sample, and then you're bombarding that in an electron microscope. and you're bombarding your images with electrons. And so this picture shows the type of raw data you get. So these little squares are highlighting diffractions or diffraction patterns of single molecules. So what's exciting is this is a single protein directly in the EM experiment. So unlike in a crystal, you don't actually get to see the molecule. You're seeing a diffraction pattern of millions of molecules all stacked together into this abstract diffraction pattern. This is actually a direct image of the arrangement of the molecules in the sample. And so the name of the game is making many, many of these measurements and then assembling them and figuring out how do they orient themselves in space. So it turns out that there's a tremendous amount of computational effort and tricks, and this part of the field is advancing hand in hand with imaging technologies, is how do you reconstruct these images? And I like this picture down here very much because it gives you a real world example of what's happening, and you're taking all of these different arrangements from what looks like a two-dimensional arrangement, and you're projecting them onto each other in 3 dimensions, giving you a reconstituted 3-dimensional view of that molecule. And so this is exactly what's done by computational processing of these direct images. And so what has happened, especially with one of this breakthrough paper in Science in 2015, is the microscope itself, the type of scope used to image, and the processing technology has gotten us to the point where it is possible to start to get these atomic level resolutions. Not quite 1.5, but it's pretty close, 2.2. And I like to point in this example. Here's that phenylalanine with nice electron density around the ring and a little hole in the middle. So that's a really nice, accurate placement of that group to give you very high resolution of single proteins in EM. And this is just a picture of some of these gigantic cryo-EMs. They're so big, they're even called Titans. And these now exist more and more places all over the world. And so these are really exciting instruments that can handle many, many samples per day. And the throughput and the sophistication is just growing year by year. And so it's really opened the floodgates on the type of structural information the field can gather. And so I put here the up to the minute plot of the growth of the field. So you can see we're almost in this exponential phase with maybe an inflection point starting around 2012, 2014. So this is when we started to see the major advances in cryo-EM. You might recall that it was the 2017 Nobel Prize, but one of the key findings was in 2015 of these super high resolution single protein structures. And so now we're seeing a database that's growing. And so the industry standard for databases seems to be separate. It's the EMDB. It's separate from the X-ray source, from the PDB. But where the PDB stands today is at around 154,000 entries, and the EM database is starting to grow. So still lags by an order of magnitude, but we're seeing nearly 15,000 structures and more coming. So this is clearly a high growth field, and it's generating a lot of excitement and a lot of intensity in the field. So the take home for you is that the structural databases are starting to expand, and X-ray is a major contributor, and so is electron microscopy and EM, to give us access to more and more information about structures. And so just a couple examples, there's so many in the literature that are exciting, but what I like to highlight here is that EM has really given us access to some of these more difficult proteins in situations that maybe were not really tractable for X-ray crystallography. So the field seems to be exploding in areas of membrane proteins, protein complexes, and any other protein that was difficult because it's so big, or difficult because getting all of the molecules in the sample to behave and generate ordered crystalline lattices. Molecules with a lot of movement really are almost impossible to get X-ray crystallography data, but turns out to be much more amenable to cryo-EM. So just 2 examples. You can find pretty much one of these in every week in the major journals. But here is a really amazing complex of a pathway to secrete a protein across a biological membrane. And in the structure, they were able to capture the machine, and in this case, a piece of the amyloid protein that secretes out of this macromolecular machine. Another example here is a machine that, again, goes across the membrane, but what this is an unfolding process, and what they've captured here is an ubiquitin molecule being unfolded and transported across a biological membrane. So the take home here is assemblies of proteins. So this one that my laser pointer is on has 3 or 4 proteins, including a multimeric transmembrane protein assembly and the substrate itself, all at fairly high resolution, high enough that you can accurately map the position of all the components, and you can even start to understand almost at single amino acid level, but certainly at peptide structural level of single peptides. So this is just one example of where the field is exploding. And so you can see, even since 2015, the growth of this field has changed the composition of the protein databases. And now we have a more insight into protein complexes and membrane proteins, where those areas were fairly rare in the crystallographic data. Okay, and I'm almost right on time, and that's the last slide. And I will now just kind of back up and open it up for questions for this segment, the final segment of the lecture from structural biology. Any questions there? It was all perfectly clear. You feel like you could run right out now and solve any structure you want? No? Silence?
I had a question actually about the proteomics section.
Please, yeah, we can go back to that. Go ahead.
I was wondering, for the reference genomes that you use, how do you deal with like potential introns when identifying new proteins?
So in other words, what if you had alternative splicing? So the linear sequence of amino acid would be defined by how it was spliced. And since there are splice variants, that could change. One answer is it's not dealt with. So in many of the studies we discovered, it's a problem. Most of the time, unless it's a study that takes into account alternative splicing, you have to go in and derive that information. When I mean you, someone who has access to that information, or you want to reprocess it. So the way you can do that is you can set up a database structure that accounts for differences in splicing. And where the field struggles goes back to a principle that we discussed very early in the lecture about coverage. So I told you over and over again that we don't have a peptide or a series of peptides that completely covers all the positions of the open reading frame. So that also means we don't have peptide level information of all possible exons. So often you don't know. You don't know if you get a peptide that's in the middle of one exon and another peptide that's in the middle of another exon, you have no idea if they came from the same exon structure. You don't know if they came from the same protein. Sometimes you can actually find peptides that span an exon boundary, and you can determine that from your data, and you can match that to the genome. So on occasion, and with effort, you can map exon-exon boundaries by the peptide that you've discovered. But it really takes using a combination of approaches and sometimes multiple databases that account for multiple possible splicing scenarios. Often what groups will do, and this is becoming more and more popular in the field, is they'll actually generate their own mRNA database. So they'll do an RNA-seq experiment, and from the mRNA, then you know how the genes are spliced, and you can derive your protein database directly from the mRNA database. So then you'll have a better sense of what is the actual diversity of exons and proteins that are expressed in your cell. But the take home is it's not often addressed, but it can be. But there is some luck involved in terms of, did you observe the peptides with high enough confidence to assign an exon? So that's a fantastic question, and it's something that we didn't even have a chance to get into in the lecture.
Thank you. That's super interesting.
Jesse, I had a question regarding what you were just talking about, how sort of labs use sort of RNA-seq data to figure out the protein sequences they should be expecting.
Yes.
I think my first question is, could you in vivo translate your RNA sequences into protein sequences? I'm saying in vivo, not in vitro. Oh, sorry. Yeah. So like
in silico?
In silico. Not even in silico.
Yes.
Even in silico, actually.
Yes.
So you could sort of have like a bunch
Yes.
of RNA-seq, sort of RNA transcription, RNA data, RNA actually, and you could translate that into proteins, right?
Yep. That's exactly what's done.
Then in theory, could you do this such that you translate the RNA, and this is, I'm saying, you're doing this in culture, you translate the RNA into your protein and mix it with the protein that you acquire from your cells, such that you can then look for low abundance proteins as well. Am I making sense?
Or yeah, generally we don't worry about abundance.
Mm-hmm.
In general, the workflows don't really take into account abundance, because you can just say that this protein should exist. And even if you interpret that from the genome database itself, what you're suggesting is use a transcriptional framework, something that you've experimentally determined, to then predict proteins. And that's absolutely true. And you could even filter that. So you have all the flexibility you want. You can make sub databases, you could filter out databases, and you could ask the question, I'm looking for a protein that's rare and of low abundance, and I would like to skew my database structure towards that sequence.
Right. So I guess my question was not exactly that.
Okay.
Slightly different. So what I was pointing at was the fact that how proteomics does not have polymerases, right? So you cannot, you sort of look at low abundant proteins effectively, correct?
Yes.
So can a workaround to that be that we take RNA, which is in our cells, you translate that to proteins in culture.
Oh, you mean, okay.
Kind of
I get it. You mean, why not take the RNA,
yes,
amplify it, and then feed that into a in vitro protein synthesis
right
mix and get more protein? Yeah, absolutely. You could do that. Yep, for sure. You're touching on one pitfall we have, and you would also have to couple that with some sort of a separation. If you could think of a way to fish out the low abundant transcripts and get rid of the high abundance, because the concept, it's big, but I'll just give you the easiest access to it. You still have an abundance problem. So when we do protein samples from cells, we are seeing the most abundant proteins. And one of the reasons we don't see the low abundant ones is that the more abundant ones interfere. There's an interference because there's more of one set of proteins. It interferes with our ability to detect the low abundance. So your idea is really clever, and it would totally work. And then I would add, if you could figure out a way to separate out the low abundant transcripts and then get rid of the high abundant transcripts, you'd even be more successful. So because you would increase that resolution in your mass spec experiment.
Right. It's like you then look at just the delta in a way.
Yeah.
Because
That would be cool. You could totally do that. You should look in literature. Chances are someone's done it. I don't want to spoil your excitement with a good idea, but it's a really good idea, and chances are there's an example of that. Probably won't be as cool as the idea you have, but I bet you it's been done.
Yep. Thank you.
That's a great idea. I like it. All right, those are great questions. All right. Well, if you think of anything else, you can send me an email, no problem. I've enjoyed this year, and I think it was all right over Zoom. I think, crossing my fingers, it'll be in person next year, but you won't have anything to do with that. You'll be off in your labs doing cool stuff. Okay, so enjoy the rest of the course. Thank you very much.
Thank you.